Welcome to SBI2. I want to thank you for kindly taking time to join us today. My name is Julie Conkright-Fincham, and I am the head of screening at the Stowers Institute for Medical Research in Kansas City, Missouri. It is my great pleasure today to introduce Jay Unruh. He received his Bachelor's of Science in Biochemistry from John Brown University and a Ph.D. in Chemistry from the University of Kansas. He completed his postdoctoral studies in the Laboratory of Fluorescence Dynamics at the University of California, Irvine, before joining the Stowers Institute as a research specialist in 2008. In 2010, Jay took the role of a research advisor, and then in 2015 became the co-head of microscopy. And since then, in 2019, he's been promoted to the co-director of microscopy, imaging, and big data, and he directs the activities for that department. If you don't know about the Stowers Institute, one of our big strengths is that we use many different model organisms for all of our research studies, one of those being yeast. And Jay is going to tell us today about GFP complementation screening of yeast internuclear membrane proteome. Please input questions in the bottom of your screen during the talk, and we will get to those after the talk. Welcome, Jay.
Hi, everyone. I'd like to take the opportunity first to thank the organizers of the meeting and the organizers of this session for giving me a chance to speak here. I'm going to talk to you today about GFP complementation methods for screening the yeast internuclear membrane proteome. Firstly, I'd like to start by acknowledging the people who did the work that I'm going to talk about here today. This was a collaboration with the Sue Jasperson lab, and in that lab, Chris Smoyer did much of this work as part of her Ph.D. project. Jen Gardner also helped with much of the strain construction that you're going to see here. And Jingjing Chen spearheaded some of the super resolution studies that I'm going to talk about at the end of my talk. Within the Microscopy Center, which is my department, Sarah Smith did a lot of the work. Brian Slaughter was involved in a lot of the discussions and brainstorming. Melania McLean did the immunogold studies that were required to validate some of the things I'm going to talk about. Zulin Yu is our super resolution expert, and he was involved in taking many of those images that I'll show later on. And Sean McKinney is an expert at microscope automation and was involved in doing those aspects of this work. On the molecular biology side, Scott McCroskey and Dan Bradford worked on automating the strain creation for this study, which was crucial to the study. So I'm going to start off with a confession. I'm not a screening person. And my role at the Institute is often to take traditional imaging assays and try to find ways to make them higher throughput. And so we utilize automation in many different ways. We utilize it for yeast transformation, tissue culture transfection, automated imaging methodologies of all sorts. And also a really important part of what we do is semi-automated image analysis. And that's often a crucial bottleneck to making traditional imaging assays higher throughput. And the bottom line that I want to communicate is that screening is no longer one specific thing. There's now a continuum of high throughput methodologies that can be applied to almost any method or question. And so that's our goal at the Stowers Institute, is to try to figure out a way to facilitate higher throughput investigations of basic science. And so today I'm going to talk to you about the internuclear membrane. And the internuclear membrane is an interesting space because, as you can see, the nuclear envelope here is composed of two leaflets. There's an inner nuclear membrane and an outer nuclear membrane. And these are one continuous membrane sheet. And the outer nuclear membrane is continuous with the ER. And so it can be very difficult to biochemically separate these two things. And these two compartments are separated by the nuclear pore complex, which I'm sure you're aware of. And what's shown here from this very nice review from the Jasperson lab is how integral membrane protein cargoes would be shuttled through the nuclear pore complex by a karyofurin, for example, which would be the standard nuclear localization sequence mechanism for entering the internuclear membrane. And so little is known about how exactly this transport pathway occurs and the processes that control the proteome at the internuclear membrane. But it's incredibly important. It's incredibly important because this is where all the chromatin is. So a large amount of gene expression regulation happens at the internuclear membrane. Also, this part of the cell controls nuclear shape regulation. And we know that nuclear shape regulation is often related to aging and cancer and other failures of this system in the cell. So control of this transport is complex and poorly understood. I already mentioned nuclear transport. It's unknown how much diffusion controls what gets from here into here. And also, once proteins are at the internuclear membrane, how are their levels controlled? It's well known that there's an internuclear membrane associated decay pathway, but are there other degradation pathways? And how do pathways such as ERAD, ER associated decay, interact with the internuclear membrane? So we want to study this in the budding yeast, and we use budding yeast for a couple of different reasons. One reason is the ease of molecular biology examination. It's very easy to endogenously see terminal tag proteins at their genomic locus in a yeast cell, and this can be fully automated, and it's fast. It can be done in a few days. There's a wide variety of growth media that can be used to select these inserts, and so this is one of the main reasons we use budding yeast. When it comes time to do N-terminal tagging, and I'm going to talk about that a little bit, we can also tag those endogenously, but we have to insert an exogenous promoter. And we're going to use the CDC42 promoter, which is a fairly conservative promoter. But you should be aware that for the N-terminal screen, some of the lowly expressed proteins will be overexpressed a little bit. These are not highly overexpressed, but they are certainly sometimes a little bit more than their endogenous counterparts. And so we're going to screen this internuclear membrane proteome with a method that's often referred to as split-GFP. Some people call it biomolecular fluorescence complementation. And it was described in this Nature Biotechnology paper in 2005. And the basic concept is that the GFP molecule is split into two parts. And we call the little part GFP 11. The bigger part we call GFP 1 to 10. And when these two pieces come together, they become fluorescent. So they're non-fluorescent on their own, and they become fluorescent when they come together. And they bind with relatively low affinity to one another. So in these control examples that you see here, we're using a protein that localizes strongly to the nucleoplasm. It isn't chromatin localized, it's in the nucleoplasm called PUS1. And PUS1 is attached to the small part of GFP, the GFP 11 part. And you can see all we have in the green channel is autofluorescent signal. And in the red channel, we have very strong nuclear localized signal. If, on the other hand, we take an internuclear membrane protein, ASI1 here as an example, and attach it to the larger part of GFP, the GFP 1 to 10 version, we don't see anything in either channel other than the autofluorescence in the green. Now, when we express these together, you can see the accessibility of this epitope at the internuclear membrane of these cells leads to strong complementation of signal. And now we see very, very strong signal here at the internuclear membrane. And this was published in a paper in JCB in 2016. And so we decided we wanted to screen for what proteins were accessible at the internuclear membrane. And we selected a little over 1,000 strains to look at, proteins to look at. We ended up counter screening 561 of those, tagged on the N-terminus. And this includes all open reading frames in yeast that are possibly transmembrane. So these are all open reading frames that have characteristic hydrophobic regions that look as though they might be transmembrane. And this was a pretty intense effort. This was a 3 to 4 month effort. It was all done with a very high sensitivity confocal microscope. This is the microscope we used. It's a Confocore 3. It has avalanche photodiodes for detection, which are incredibly sensitive. This allows us to approach single molecule sensitivity, but the sacrifice is speed. This is a fairly low speed process. And we did all of our imaging by hand, and we did all of the annotation by hand as well for this first round of screening. And so let's talk about some of the results. So we had three basic categories of results. We had proteins that didn't show anything at the internuclear membrane, so clearly negative for at least that part of the protein at the internuclear membrane. We had proteins that were strongly positive for the internuclear membrane. And then we also had proteins that were positive at the inside of the nucleus in the nucleoplasm, but were soluble, were freely moving throughout the nucleoplasm. And this is expected because even though we tried to choose proteins that were transmembrane, some of them were characterized as transmembrane and ended up not being associated with the membrane. So to summarize our results, we had 529 strains in our initial screen that were negative. We had 169 that were soluble and positive, and then 312 that were positive at the internuclear membrane, which is what we were trying to screen for. Then we did a secondary screen with these negative hits. The C-terminus of many proteins has signal sequences that are responsible for localization within the cell. And so we wanted to counter screen those tagged at the N-terminus. And once we did that, we found 100 more proteins that were positive for the internuclear membrane. And so we have 412 proteins that are potentially at the internuclear membrane. And given that high number, we were somewhat skeptical of those results, and we wanted to do strong validation of these results. And the strongest way we know how to validate is with immunogold electron microscopy. And so you can see here in electron microscopy, you can easily resolve both the inner and the outer leaflet of the nuclear envelope. And these black dots are immunogold signals, and we can quantify what percentage of those are present at the internuclear envelope. And you can see that some of them are quite low, but many of them have significant numbers of immunogold localizations that are at the internuclear envelope. So we were able to confirm many of these signals. Another way we validated this was with theta nuclei. Theta nuclei are so named because they look like the Greek symbol theta. And these are nuclei that have a bridge of internuclear membrane that goes through the middle of the nucleus. And so we don't expect any outer nuclear membrane proteins to be on this bridge. And so if we see signal on that bridge, as you can see here, we see it here, and we see it here, we were able to validate the localization of many of our signals to the internuclear envelope. And so we created a tool, a website that you can access if you want to. If you go to this link, you can look up your favorite protein and see if it was screened in our screen and whether or not it was at the internuclear membrane. And this is a tool for the community to use. This is showing the C-terminus viewer, but there's also an N-terminus viewer as well. So the big question we had then at that point was, why are so many proteins at the internuclear membrane? And we don't really know. One of the things obviously the most intriguing option is that these have unknown novel functions at the internuclear envelope. Like I said before, we don't really know what role diffusion and nonspecific transport plays. But in particular, we were interested in the internuclear membrane quality control process. And so what we did was we did a counter screen where we deleted different pathways of different quality control components within the yeast cell and saw how that affected our internuclear membrane localization. And so the particular one we were interested in was the ASI1, which is the internuclear membrane associated decay pathway. Some of these components are shared between the ER and the internuclear membrane, but we also had components that were unique to the ER decay, and PEP4 is a vacuolar decay component. And so what we did was we started a second round of screening, and now since we want to screen so many more proteins, we wanted to try to automate things and speed them up a little bit, so we didn't commit so much time to this. And so part of this was we just had to eliminate some of the more difficult strains to grow and to image. We switched our microscope now. We're using a spinning disc microscope with an SCMOS camera, scientific CMOS camera. This is very sensitive, and it has a wide field of view for fast screening, but it is not as sensitive as our first round of screening was. And so, but it's able to see many of the proteins that we're looking for. We're also using, this is a Nikon microscope, we're using their jobs interface for automation, and that allows us to automate many of the imaging processes. And all of this together allowed us to image 96 strains in 2 hours. And so altogether, we were able to do all of this in less than a month of imaging time. So a considerable speed up compared to what we did before. The other thing we've done is we've used automated segmentation and analysis, image analysis, and that has allowed us to very rapidly analyze these results and do them in a quantitative fashion. So our results fall into 3 basic categories. One is proteins that were not at the internuclear membrane and suddenly are. And this, I'm showing the example here of ASI1 deletion. That was that INMAD component. A more common occurrence is a protein that was weakly at the internuclear membrane and becomes stronger in that compartment. And then we had some that went from basically negative or weak soluble signal to stronger soluble signal. And we're not going to talk about those so much. So I'm not going to go into incredible detail on these results, but I wanted to talk about just general trends that we saw. And one of the general trends that we saw was that this ASI1 protein deletion results in a much larger proportion of proteins increasing at the internuclear membrane compared to some of these other categories that we have. What's interesting, though, is, and that makes sense with its role as an INMAD component. What's interesting, though, is that all of these components affect the localization of significant numbers of proteins. But in addition, there's many of the proteins at the internuclear membrane that are not affected by any of these pathways. So a couple of conclusions we can draw off from that, just broad conclusions. The internuclear membrane proteome contains a large number of proteins. We are still not sure whether these are functional or accidental components, but these are fully formed proteins. And so it's likely that even if there's accidental diffusion of these proteins into the internuclear membrane, they may be functioning in a non-canonical fashion once they're there. Many are unaffected by degradation pathway mutants, so we don't know if there's other undiscovered pathways that are involved, or potentially these pathways work together in compensatory ways that we haven't anticipated yet. And as I said, the INMAD complex results in the highest proportion of INM increases as expected. So I wanted to spend the end of my talk talking about some other fun things that we're doing with split-GFP. And so I want to start with a question that came up as we were doing this screen. So one of the things we noticed was at the internuclear membrane was the ER translocon complex. And this is involved in inserting newly translated peptides into the ER membrane. And this complex has 2 versions. It has a version with a core of Sec61 and a version with a core of SSH1. And these are both at the internuclear membrane in high quantities. What's interesting is these proteins form complexes with SBH1 and SSS1, and in this case, SBH2 and SSS1. And SBH2 is at the internuclear membrane, as you can see here, but SBH1 is not at the internuclear membrane. And so that raised a question as to how this complex can potentially function at the internuclear membrane. Is SBH2 somehow substituting for SBH1 in this complex at the internuclear membrane? So in order to investigate this, we turned to a technique called fluorescence cross-correlation spectroscopy. Fluorescence cross-correlation spectroscopy measures the noise in the co-diffusion of labeled molecules in order to measure the extent of co-diffusion. And so here, as an example, you could park your confocal microscope somewhere in a cell, and if you have green and red molecules diffusing independently of one another, you're going to see random signals in their diffusion traces. But if you have molecules moving together, you're going to see that these signals are highly correlated, and that allows you to measure the extent of interaction of these proteins. On the membrane, we have to do a little trick We scan repeatedly as a function of time, and that allows us to track the membrane. Here's those scans in a chymograph form. We can track the membrane while we watch molecules diffuse and fluctuate on that membrane. Okay, so getting back to our Sec61 SSH1 complex, we started by looking at the cortical ER, where we expect these to be behaving in their canonical state. And as expected, Sec61 has a high affinity, has a low KD for SPH1 at the cortical ER. Conversely, SSH1 has a low KD or a high affinity for SPH2, as expected. And when we go to the internuclear membrane, the affinity of both of these components for SPH2, the only component that's at the internuclear membrane, both of these have a very high affinity for that component, indicating that it's quite possibly substituting in where the other component is not available at the internuclear membrane. Another fun thing that we've been able to do with split-GFP is we've been able to utilize it in super resolution studies to look at the geometry of different complexes. One example of that is our investigations into the sun-cache complex. The sun-cache proteins are known to span the nuclear envelope and act as tethers for the nuclear envelope. And in yeast, the proteins that do this function are MPS3 and MPS2. And these proteins also are involved in stabilizing the centrosomal pore in the nuclear envelope. Yeast has a centrosome that sits in the nuclear envelope called the spindle body. And we wanted to understand the geometry of this interaction. And we had a hypothesis that this interaction at the C-terminus could be added to an interaction at the N-terminus that could possibly stabilize the curvature of the membrane at the nuclear envelope. And so we did super resolution with this split GFP, where we put the split GFP either at both C termini of MPS2 and MPS3 or at both N termini. And we get complementation in both cases, confirming the possibility of this parallel complex forming here. The other thing that was interesting is we also see complementation of this at the duplicated centrosome. And this duplicated centrosome hasn't inserted into the membrane yet. And this suggests that potentially MPS2 and MPS3 are involved at the duplicated centrosome, starting to remodel the membrane in preparation for insertion of this new centrosome. And that's an intriguing possibility that we want to pursue further. So to conclude, GFP complementation assays enabled our proteome screen. We saw a very large number of transmembrane proteins at the internuclear membrane and are still investigating that. The quality control pathways influence some of those localizations, and INMAD clearly has a large role, but there's many also that aren't involved. We were able to use GFP complementation for fluctuation microscopy, spectroscopy of internuclear membrane interactions. And then finally, we could use GFP complementation with super resolution microscopy to investigate structural components in situ. So I'd like to thank you for your time, and mostly I want to thank Jim and Virginia Stowers for their incredible generosity, donating a large amount of their wealth to make the Stowers Institute's mission possible and to allow me to do the collaborative work that I'm able to do at the Stowers Institute. Thank you.
Wonderful talk. If anybody has questions, please put them into the question box at the bottom of your screen. The first question that we have is, what controls or counter screen did you incorporate for the lack of reversibility of the GFP by BIFC once formed?
Yeah, that's a really good question. So the BIFC molecule that we're using is one, I put the reference to the Caventus paper in that slide. And so if you can look back at the slides, you can go see that. But in particular, this split GFP molecule is reversible. How reversible it is depends on time scale, and I believe the dissociation constant is in the micromolar regime. And so we do think that we're probably stabilizing by a slight amount some interactions, but the correlation that we see with the immunogold and with the theta nuclei, which don't have this interaction on them, suggests to us that we're not dramatically influencing the fraction of a molecule at the internuclear membrane.
The second question is split-GFP fluorescence retained after fixation with formaldehyde or other cross-linking agents?
Yeah, that's another great question. So I don't know how thoroughly we've tested that. I know we saw some changes upon fixation, and so we ended up doing the entire screen with live cells. But it would be interesting to explore that further, especially for higher throughput screening.
A third question is, how do you think the structure of the nuclear pore complex influences the composition of the internuclear envelope?
Yeah, so that's a really good question. So if COVID wouldn't have happened, I would be probably talking about that today. We've been using super resolution with split-GFP to try to understand the structure of the nuclear pore complex. And so we don't have a complete answer to that, but we're definitely working on tools to more thoroughly explore the structure of the nuclear pore complex and try to understand how proteins use that to access the internuclear membrane and to leave the internuclear membrane.
I have a question for you. So since this is a reversible complex, and you can see almost at a single cell resolution, are you able to measure residence times of the protein-protein interactions?
We haven't done that, but I think that's a great idea. It would be really cool to look at these INMAD deletions and see if that changes the residence time. So I think that's a great idea, but we haven't done it yet.
Okay, we have time for just a couple more questions. How would you apply your technology to other cell types, for example, mammalian cells?
Yeah, so the struggle we have with mammalian cells is that we have this +1 molecule that is nicely nucleoplasmic and basically has zero concentration in the cytoplasm. And so that allows us to interrogate only the internuclear membrane. And in mammalian cells, that's harder to generate. And so we're looking at a lot of different options for trying to understand what goes on in mammalian cells, but it's an area of active investigation.
Great. There's a question of, is GFP fluorescence reconstitution here happens due to two proteins co-localizing or co-localizing and interacting as well?
Yeah, so this is interaction here. But the co-localization that I talked about when I talked about fluorescence cross-correlation spectroscopy, that was co-localization. That was actually tripartite co-localization. So it was the complementation of the GFP plus co-diffusion with another protein that we were looking at.
Thank you very much. Jay, I want to thank you for a wonderful presentation and everyone for listening. Take care.
Thank you.